Uh, Good morning to all of you. I hope you can hear me and see me. In the last class, if memory goes right, because last week I couldn't engage because I was in the practical examination work. Before continuing the immunology, I would like to rebrush what we have discussed in the previous class. Uh, we started our class in order to find out answer for certain questions. Why all are not having the same level of protection against the pathogen? How the vaccination helps us in protecting us from the diseases? Uh, then there are certain diseases, once we encounter it, will provide lifelong immunity. Whereas there are certain diseases, there is no guarantee that within a month also it will reoccur. So we have tried to we have started with these questions, and then I told that by studying the immunology, we will be able to find the answer for this. So in the immunology, in the very beginning, I have started with some of the glossaries or the terminologies that we use very frequently in the future discussion. Today we will continue from there, and I will also introduce few more things even today. So we have seen what is immunogen. Immunogens are nothing but the antigens. But remember, all antigens are not immunogens. So uh, because there are some are very small molecules called haptons. They bind to antibodies or sometimes T cell receptors, but in spite of that they cannot initiate the immune response. Otherwise, an antigen is the one which has to initiate the immune response. So now the immunogen, what is the exact difference between immunogen and antigen? Many times we use the term or we consider immunogens and antigens as synonymous, though at a gross scale it is true, but at the certain level, if you observe, there is some difference. An immunogen is a stimulus actually that produces a cell mediated immune response. Cell mediated immune response or humoral immune response, what we but whereas antigen is also a substance that binds specifically to the antibody or to the T cell receptors. So antigens bind specifically to the antibody, but immunogen is a stimulus, and because of this stimulus, there will be uh, the the cell mediated responses will start. Then we have also one more term, epitope. What is this epitope? Epitope is a portion of the antigen. Uh, this is a very important thing. I'm going to discuss about that in detail because it recognizes, not only recognizes and binds with uh, antibody or T cell receptors or major histocompatibility complexes. So that's what we call epitope. Then one more term, the hapten. Haptens are low molecular weight uh, substances that can be made out of the can be made immunogenic by conjugation with suitable carriers. Haptens on their own, they are very low molecular weight molecules, so that's why they cannot initiate any immune response. But they can initiate immune response provided they attach with some suitable carriers, and most of the time these suitable carriers are nothing but the proteins. So that's the then we have one more term, that the paratope. Paratope is the site or a location on the variable domain of a antibody or T cell receptors. Uh, T cell receptors, and that bind to the epitope of an antigen. Actually, epitope and paratope, they are compatible. Epitope, epitope is found on the antigen and paratope is found on the antibody. I will come to that later. See here, this is the T cell receptor, this is the peptide, and this is the MHC, that's major histocompatibility protein, complex protein, and these are the T cell receptors. And here, the epitope, they can bind here. I will come to that. Now, talking about the antigens, to become an antigen, uh, a, ma- any material should have certain properties. That is the features or characteristic features of the molecule that determine the antigenicity and immunogenicity. What is that? It should be able to have four important properties. One is foreignness, second one is the molecular size, then the third one is chemical structural complexity, and fourth one it should have a epitope. Then only any material can become an antigen. What do you mean by foreignness? In general, remember, these antigens are foreign compounds. They are non-self. Molecules recognized as self are not immunogenic. Suppose if you take anything, for example, you may wonder, you eat lot of things. They are all foreign material. In spite of that, they do not cause any harm or they do not cause any immune response in our body because that food molecules that we eat, our body recognizes it as self, and self molecules are tolerable. But a material or anything to become antigen, it must be recognized as non-self, that is foreign. So most of the pathogens that enter our body, they are treated as non-self or pathogen. So that's why they can initiate what you call this uh, immune responses. That's the body defense mechanism. The second one is the molecular size. To become an antigen or an immunogen, the protein should have high molecular weight, that is above 1 lakh Daltons. Generally, molecules with molecular weight below 10,000 are weakly immunogenic, and very small ones, small example amino acids and certain small molecules, uh, they cannot become immunogenic unless they are linked with a carrier protein. And any material, any small molecular weight compound material, even if it becomes, when it is linked with a carrier protein, it can initiate immune response, and those materials are called as haptens. Now, the chemical nature, chemical complexity. So remember, proteins and polysaccharides are most antigen. To become a, generally the antigens are either proteins, or most of the time they are protein, or sometimes they are polysaccharides. Lipids and nucleic acids are less antigenic. Most of the time they do not initiate any responses. Uh, but the even lipids and nucleic acid become antigens provided they are in combination with certain protein, which enhances the antigenicity of the from the lipids and nucleic acid. So to become an antigen, certain amount of chemical complexity is also very, very essential. Uh, for example, amino acids, which are the basic or the uh, fundamentals of a protein, uh, generally they cannot. But uh, amino acids, if they have homopolymers, they are less immunogenic. But if they are heteropolymers, that means heteropolymer means containing two or more different amino acids. If there's only one type of amino acid joined together, they become less immunogenic. Suppose if there are heteropolymers, two, three different types of amino acids joined together form a polypeptide chain, then they become uh, more immunogenic, I should say. Then antigen determinants. uh, on the antigen, there must be some determinants, and these determinants are nothing but small chemical groups, and they are popularly called as epitope. And these epitope, uh, Actually, they elicit and react with antibody and progresses the immunogenic responses. And an antigen may have one or more epitope. Most antigens have many epitopes. So that's why most of the antigens are multivalent. If there's only one epitope, we call it as univalent. If there are two epitopes, we call it as bivalents. And if there are more than uh, two determinants on the antigen, they are called as multivalent. See, this is the how antigen and see this is the antibody. This is the antigen. See, on this there are receptor sites. These are known as epitope, and they bind with the antibody. See here, this is what is the paratope. Actually, receptor sites are known as paratope. So this epitope is the active site. We can say that active binding site that is found on the antigen, which have certain level of chemical complexity. See, these are all uh, antibodies, and these are the antigen molecule. On that, you can see the epitopes. Epitopes. So antigen receptors will be like this. I'm not going to the details. See, different types. Uh, this is a monovalent. This is the bivalent. It is a multivalent. If there are more epitope, we call it as multivalent. If two bivalent, if there's only one epitope, we call it as monovalent. Then uh, haptens. Haptens generally they are called as incomplete antibody because this is a molecule that cannot induce immune response by itself unless uh, they are with some carrier protein. But these hapten molecules can induce immune response by itself, cannot induce immune response by itself, but they can react with specific antibodies. Though they can bind with specific antibodies, they fail to elicit the immune responses unless they are combined with larger molecule, normally proteins. And those proteins are called as carrier proteins. These haptens are usually small. Many drugs, either penicillin, most of the penicillin and antibiotics are haptens. And another one, catechol in the plant oil, um, that causes poison, oak and poison EV. The poisons which are found either in the oak plant or EV plant, they are all haptens. They cannot induce reactions unless they are combined with some larger molecules such as proteins that the carrier. See the haptens, uh, they can enter into the body, but no antibodies are produced. But when they are with the larger one, they can produce. See these photographs. So a typical antigen antibody reaction should happen for any of the immune response to happen. Uh, for example, if you take a bacteria, gram-negative bacteria pathogen, may have several antigens on that, or immunogens like flagella, pili, cell wall. All of them function as an antigen on that surface. This is the antigenic structure of a bacteria. See here on the epitopes on it. This is the antigenic structure of a virus. A viral this thing. Here these are the haptens. So usually these antigenic properties of bacteria or toxins or virus is used in practice of reproducing artificial immunity against the infectious disease. By in vaccination, they are used. When we discuss vaccination, there we will discuss about it. Then one more thing, when the antigenic structure of the host are similar to those of the causal agent, then these microorganisms are incapable of producing immunity. Suppose if an antigen enters our body, a foreign pathogen is generally considered as an antigen. And if the structure of the foreign antigen is similar to that of the host antigen structure, then this pathogen is incapable of producing the immunity. Um, as a result, what happens? Immunity, if the immunity is not produced, the disease follows a grave course, very severe it will be. Uh, Only we can protect such people from vaccination. For example, the human erythrocytes have antigen in common with the streptococci. The antigens of the human erythrocytes, human RBC, and these staphylococcus and streptococcus, these are the bacteria or E. coli, salmonella. They have the similar one, and such condition is called as antigen mimicry. Actually, antigen mimicry. Uh, the pathogens mimic the antigen of the host. As a result, the immunity is not conferred to the host. So, as a result, the disease will be more severe. Then one more type, that is the isoantigens. Uh, these are the substance which have antigenic properties, but they are contained in some individuals of a given species. In not all individuals of a given species, but they are found only in some individuals of a species. For example, isoantigens in the WBC, blood platelets, lymphocytes, uh, mitochondria. Such isoantigens have been revealed in some individuals of the species. Um, these isoantigens may vary. For example, if you take the human beings in the RBC, we have basically two different types of antigens, A antigen and B antigen. Based on this antigenic structure, the people are subdivided into four groups, A blood group, B blood group. Where both are there, we call it as AB blood group. When none of them are there, we call it as O blood group. Um, these are because of the isoantigens. Like isoantigens, we have autoantigens also. These are the substances which are capable of immunizing the body from which they are obtained. They are the self antigens. In other words, these are the antigens, self antigens, which are present in our own body. Generally, they are non-antigenic. That means they do not produce any problem or immune responses, and not normally found in free circulation also. And they are not recognized as self-antigens also. But they have the capacity of becoming modified and capable of bearing these antigenic functions. Such normal autoantigens are found in the lens of the eye, in spermatozoites, in seminal gland and skin, in the emulsions of kidney, liver, lungs, and other tissues. You see autoantigens. Under ordinary conditions, they do not come in contact with the that is immune system of the body. Therefore, antibodies are not produced against such cells and tissues. Suppose if these cells or tissues are injured, which are having this autoantigen, then autoantigens may be absorbed and may cause the production of antibody, and which may have the toxic effect on the corresponding cells. Because our own defense system will start fighting against our own cells. This is the one. Uh, Autoantigens are usually under the influence of cooling, radiation, drug, sometimes virus infection, or autolysis, and other factors are responsible for the origin of this autoantigen. And most of the allergy that we have is because of active autoantigens, where they start immune response. Then one more group of antigens, that is histocompatibility antigens. Uh, these are the antigens present on the cells of each individual species. These are some specialized ones which are found on the cells of each individual of species. These are proteins and also called as alloantigens if they differ among the members of the same species. If they differ among the members of the same species, they are called as alloantigens. Um, Usually, these antigens are encoded in our genes, known as histocompatibility genes, and collectively they constitute a complex known as major histocompatibility complex. This major histocompatibility complex, in short, which is known as MHC, are located on the short arm of chromosome number 6 in case of the human being. Major histocompatibility complex is HLA, human leukocyte antigen. Um, this plays a very important role in the organ transplantation. Actually, if suppose if there's a kidney transplantation or heart transplantation or liver transplantation takes place, uh, the success of this transplant depends on HLA, recipient's HLA, the person who receive it, his HLA, that is human leukocyte antigen. If the HLA protein on the donor cell differ from those of the recipient cell, the immune response will start and graft may be rejected. That's the case because of HLA, human leukocyte antigens. If they are different in the host as well as in the recipient, then the organ or the tissue that have been grafted will be rejected. There are MHC proteins, class 1 MHC protein. These are the glycoproteins found on the surface of all nucleated cells. That's a small typing mistake. Class 2 MHC protein, these are the glycoprotein. These are found on the certain surface of the certain cell, especially on macrophages, B cells and all. But the biological importance of MHC is the, uh, as I have told you, for example, uh, in the graft, when there's an organ or tissue transplantation, because they recognize the antigen is depending on the association of antigen with either class 1 or class 2 protein, and they decide whether to allow them, whether it's a self uh, or non-self, and if the MHC consider it as non-self, the graft is rejected. So these are some of the importance. So this is about the antigens. In general, antigens are the one which are considered as the pathogens, which enter our body and causes the immune response. Then comes the antibodies. What are antibodies? Antibodies are also proteins. Mostly they are proteins that recognizes and bind to a particular antigen with very high specificity. Basically, antibodies are produced in our body by our immune system to nullify the entry of antigens, because once the antigen enters our body, antibodies are developed in our body by the mm, cells, and these antibodies are proteins and they recognizes and bind to a particular antigen. They are highly specific. For a specific antigen, there's a specific antibody. So they recognizes and bind to a particular antigen with highest level of specificity. Antibodies are formed only when our immune system or the cells or body are exposed to antigen. Unless our body is not exposed to antigen, antibodies are not formed. Remember, even in the present scenario, you know, in the COVID-19 also, once you get the infection, antibodies get developed. That means, well, only antibodies will develop only when the COVID-19 virus enter our body. Otherwise, no. Each antibody has at least two identical sites on which the antigen binds. and the sites on this antibody where the antigen binds is known as antigen binding sites. So that is known as valence of an antibody. Number of antigen binding sites on it. Most are bivalent. There are two antigen binding sites, but some may be univalent, some may be multivalent also. And these antibodies belong to a group of serum proteins called as immunoglobulins. These are all serum proteins because antibodies are proteins. They are produced in the blood serum and uh, they are popularly called as immunoglobulins, in short, IGS, immunoglobulins. So antibodies are nothing but the immunoglobulins. There are five classes of antibodies in the human being. There are five classes of antibodies. One is antibody G, antibody M, antibody A, antibody D, and antibody E. So these are different types. So they are also called as immunoglobulins, and each immunoglobulin has a specific structure. Mostly the immunoglobulins are having Y-shaped structure. Immunoglobulins are having Y-shaped antibodies or immunoglobulins. See, all of them are showing a Y-shaped structure. And I will come to that. An antibody or immunoglobulin, if you see, they are glycoproteins. Proteins basically sometimes attached with the carbohydrates, so that's why they are called as glycoproteins. They are glycoproteins and they are made up of four polypeptide chains. Any antibody you take or immunoglobulin you take, they are made up of four polypeptide chains, out of which two are light polypeptide chains, they are alpha chains, two are heavy polypeptide chains, they are beta chains and designated as H. The heavy chains are designated as H and light chains are designated as L. All these four chains, two light chains and two heavy chains, they are linked by a disulfide bond. Just a disulfide bond. And the terminal portion of this light chain has antigen binding site, that is the paratome. Terminal portion of this L chain contains the antigen binding site. The H chains are distinct for each of the five immunoglobulins. I have told you there are five classes of immunoglobulins. These heavy chains are distinct. Each class of protein has their own type of H chain. And terminal portion of H chain also participate in the antigen binding sites. Even they have FC fragments. There's a carboxyl fragments on them. See, there are four chains, two light chains and two heavy chains. These are heavy chains. These are light chains. And heavy chain and light chain, which you can see. And in the terminal end, there is an antigen binding site, which you can call as paratope. See the light chain, light chain. These are the heavy chain, and all these chains are joined by means of disulfide bonds. So antibody molecules are composed of two identical heavy chains. See, this is one and this one. Two identical heavy chains and two identical light chains. See, and in this they have two regions. One is the B region and the variable region, and another one the constant region. This is the variable region, antigen variable region. This is the region where actually the antigens bind. So they are called as variable regions. But these are constant regions. Only the terminal free ends are variable regions. Base, it's designated as C or constant region. So that's why each H chain and heavy chain and each light chain has variable region and a constant region. Usually the variable region lies in the terminal portion of the molecule. This is the V region. Variable at the terminal portion of the molecule. And why it's called as variable region? This region varies from individual to individual or from species to species with respect to the amino acid sequences. And this variable region is very important because that is the region responsible for antigen binding. C region lies on the carboxyl terminal portion and unvarying amino acid sequences. This region, they are constant region. They do not show any variation. It is responsible for biological function. They do not bind with the antigens. That's the structure of an immunoglobulin. So immunoglobulin classes first I have told you. Immunoglobulin G because 80% of the antibodies we have is immunoglobulin G. It's a monomer. 80% of our serum antibodies, blood serum antibodies are and immunoglobulin G. They are found in blood, lymph and intestine. Their half-life is 23 days. They have certain characteristics. One is complement fixation. They can fix with the specific antibody antigens. Even these antibodies show placental transfer when from the mother to fetus through the placenta. Uh, the important function of this immunoglobulin G is enhances phagocytosis, cell eating process. They also neutralizes the toxins and virus. As they can pass from mother to fetus through the placenta, they protect the fetus and newborn from the pathogens. So, antibody classes. See, this is the structure of immunoglobulin G. There's a disulfide bonding, and this is the light chain, this is the heavy chain, and these are the variable regions here, though they are not shown. And sometimes they attach with FC fragments. See, this is the antibody binding site, but heavy, the constant regions are having biological activity, but the variable regions are mainly concerned with the attachment with the antigen. Next one is, yeah, this is the same one, IgG, immunoglobulin G, highest concentration. Um, there are again four types: immunoglobulin G1, G2, G3, and G4. We need not go to that details. If you see the structural feature of an immunoglobulin, uh, two H chains and two light chains. Each H, the heavy chain is having around 50 kilodaltons molecular weight. The light chains are having only 25 kilodalton. Of course, Fabian will be not. It in, usually, the immunoglobulin G concentration in the blood serum increases. Suppose we have chronic granulomatous infection. If you have chronic infection, even infections of all types, hyperimmunization, when we are more immunized, then also it will be there. Especially in liver diseases, immunoglobulin G will be more. Even if we are malnutrition, deficiency of nutrients, then also there. Uh, most of the hypersensitivity and all those things, immunoglobulin G will be there. But the immunoglobulin G will also reduce in agam- anemia. This is the condition where lowering the there is globulinemia is a condition where immunoglobulin G concentration is less. It happens in lymphoid aplasia, or when there's the immunoglobulin A deficiency, it is also seen immunoglobulin G deficiency is also there. Or sometimes immunoglobulin A myeloma cancer or proteinemia. There are so many conditions when they so this immunoglobulin G, though there are four chains or two reasons, heavy and light chains, fragment antigen binding, where it takes by means of the disulfide bond, even we can separate. That's also possible. So light chains, heavy chains, the same thing which I have discussed. I will go to the next one. And immunoglobulin G antibodies are generally larger antibodies, about 150 kilodalton, and they are heavy chains and light chains, both of them are there. So uh, this is about the structure of the uh, any antibody. Generally they are, as I have told you many times, they have been, it has been asked for uh, examination also, the structure of the antibodies. So the structure of the antibodies, when asked, it is better to write more about this immunoglobulin G, which has Y-shaped structure, two light chains, two heavy chains, variable regions, constant region, and they are bound by what is known as the disulfide bonding. So this is what you are expected to uh, write. And in the for the day, I will stop here. And in the next class, we will continue. I may require another 1.5 hours to complete this. Uh, Especially, uh, we have to discuss something about vaccination also, and antibody antigen reactions. Or even if the time is there, even we can discuss even now also, because there is not much to discuss. So now, uh, antigens and antibodies. ಹಲೋ, ಕೇಳಿಸ್ತಾ ಇಲ್ಲ ಅಲ್ವಾ? ಇಂಟರ್ನೆಟ್ ಹೋಯ್ತು. ಹ್ಮ್. ಹಲೋ, ಕನೆಕ್ಟ್ ಆಗಿದ್ಯಾ? ಕೇಳ್ತಾ ಇದೆಯಾ? ಹಲೋ, ಹಲೋ, ಹಲೋ. ಅಲ್ಲಿ ಕರೆಂಟ್ ಹೋಗಿ ಇಂಟರ್ನೆಟ್ ಹೋಗಿದೆ. ಈಗ ನಾನು
Yes, sir.
ಮಧ್ಯದಲ್ಲಿ ಕನೆಕ್ಷನ್ ಹೋಗಿತ್ತಲ್ಲ, ಅಲ್ಲ ಕೇಳ್ತಾ.
Yes, sir.
ಓಕೆ. ಮತ್ತೆ ಈಗ ನಾನು ಕಂಟಿನ್ಯೂ ಮಾಡೋದಿಲ್ಲ. ಯಾಕೆ ಅಂತಂದ್ರೆ ಮತ್ತೆ ಈಗ ಇಂಟರ್ನೆಟ್ ಇಲ್ಲ ಸರಿಯಾಗಿ. ನನ್ನ ಮೊಬೈಲ್ ಇಂದ ಕನೆಕ್ಟ್ ಮಾಡಿದ್ದೇನೆ, ಆದ್ರೆ ಯಾಕೋ ಸರಿ ಇಲ್ಲ ಸೌಂಡ್ ಇದು. ಸೊ ವಿ ವಿಲ್ ಮೀಟ್ ಇನ್ ದ ನೆಕ್ಸ್ಟ್ ಕ್ಲಾಸ್. ಓಕೆ.